All right, so let's talk about DNA and then gene expression. So one question came about is, how do we know that it's DNA that's the genetic material? If you remember, in the last lecture series, I talked about T.H. Morgan's experiments with fruit flies that established that chromosomes are where the genetic material is. However, chromosomes are made up of protein and DNA, and it was not at all clear initially that it would be DNA that was the genetic material. DNA was thought to be too simple, the structure wasn't known. Protein, though, had 20 different amino acids and could form an endless variety of forms. So many scientists thought that it was going to be protein that was the genetic material, and that DNA was just going to be the scaffold that our genome was on. So how was that figured out? And we're going to talk a little bit about DNA replication and mutations. So T.H. Morgan, with his white-eyed fly mutant, showed that genes are in fact on the chromosomes because they showed that the white-eyed fly allele was in fact on the X chromosome, with male flies having only one and females having two, and that being the only real explanation for the inheritance pattern that they saw with that. So which was it, DNA or protein, that was in fact the genetic material in chromosomes? DNA was thought to be too simple, and both things, DNA and protein, were present in a variety of organisms that had been studied, from bacteria to eukaryotes, and also viruses that infect those cells also had protein and DNA. And which we now know that some viruses have RNA instead of DNA, but that was not known at the time. Okay, so there were several experiments that seemed to establish that DNA, in fact, was far more likely to be the genetic material than protein. But it wasn't until around 1952 when Alfred Hershey and Martha Chase published their experiments that people were finally convinced. Because you know, when the thing that turns out to be the most likely explanation is not what people expected it to be, for extra proof is required to establish that. And that was the case here as well. There were other experiments before the Hershey-Chase experiments that showed that DNA was the genetic material, but because that was not the expected outcome, it took more experiments to show that. And the Hershey-Chase experiments are still considered today to be one of the most elegant proofs of concept in biology. So they took advantage of the fact that bacteriophages are viruses that attack bacteria, and we can separate bacteria from their phages pretty easily. This is an electron microscope image that was, oh wait, I need my pointer. This is an electron microscope image that was, of course, made far later than this. And this is just a little tiny piece of a bacterial cell here. And the little lollipop-like structures on here, those are bacteriophages. And we now know how these work, that in fact the phages, which contain protein on the outside, they have a protein capsid, and then they have DNA on the inside. They're DNA viruses. That the protein stays on the outside of the bacterial cell, and these phages inject their DNA into the bacteria. So we know that now. So Hershey and Chase took advantage of the fact that you can grow phages in different populations with E. coli, and use radioactivity to label different substances. And they took advantage of the fact that protein contains sulfur, but DNA does not. And DNA contains phosphorus, and protein does not. So if you use radioactive sulfur and radioactive phosphorus and have bacteria and phages growing with one radioactive element but not the other, you can label either the protein or the DNA with radioactivity. And that was a fantastic idea to do that. Okay, so you grow because viruses have to grow on a living cell. You can't just grow viruses in isolation. You have to grow them on something. So they grew their phages with bacteria, and the amino acids that were supplied to the bacteria for growth and thus would end up in the phages were labeled with radioactive sulfur. So you can see S35 there, that's radioactive sulfur. So protein contains sulfur in it, so we end up with radioactively labeled protein capsid on the viruses. They had another population of viruses that they grew on bacteria that had radioactive phosphorus in their growth media. And phosphorus is not found in protein. You can see that P32 there, that's radioactive phosphorus. So these ended up with radioactively labeled DNA. DNA contains a lot of phosphorus, but no sulfur. Protein contains sulfur, no phosphorus. So then you separate your radioactive viruses from their bacterial growth media, and then you infect clean bacteria with them that are not radioactive. So they took their E. coli, clean E. coli, and infected them with the two phage populations separately, of course, because we want to see what happens separately. And they left them mixed together just long enough for infection, but not long enough for replication. So about 20 minutes. And then with agitation and centrifugation, they separated the phages and the viruses. And then they asked in the bacterial cells, our E. coli that are left here, and of course these were E. coli bacteria. E. coli are the workhorse of the molecular biology lab. They asked which bacteria are radioactive, thinking that whatever was necessary to pass on the genetic material of the viruses, that would be inside of the bacterial cells. And in fact, they found that the bacteria that had been infected with the P32 labeled phages, in fact, were now radioactive. So showing that only DNA and not protein was transferred from the phages into the bacterial cells. Now, if they had used viruses that infect animal cells, this would not have worked, because normally when viruses infect animal cells, the entire virus particle, protein and all, ends up inside the cell. So this worked very well with these bacteria and their phages, but it wouldn't have worked with any viruses. Many viruses don't operate this way. But it worked very well. And this was finally the final evidence that you don't need a protein to transmit hereditary information. You only need DNA. And remember again, this wasn't the only experiment that showed this. This was one of the experiments that showed this. So around the same time, another guy, Erwin Chargaff, was doing experiments with just looking at the gross amount of different bases that we find in DNA in different species. And so we use A, T, G, and C to represent those 4 bases. And what he discovered is that the exact amount of each of these bases varied and was species specific, but the amounts of A and T were always the same, and the amounts of C and G were always the same. And they did not understand why this would be the case, but the total amount varied. And they weren't always the same. So A is for adenine, G for guanine, T for thymine, and C for cytosine. And if you remember back to our biochemistry, these are just part of a nucleotide, these 4 bases, which we call A and G are purines, and T and C are pyrimidines. And they're one of the 3 main parts of a nucleotide. We have a sugar, which for RNA is ribose, and for DNA is deoxyribose, and at least 1 phosphate. But there could be up to 3 or occasionally even more than 3 phosphates. So how these fit together was not figured out until after the Hershey Chase experiments were completed. So once it was figured out that DNA was the genetic material, there was an enormous race between different labs to be the first lab to figure out the structure of DNA, because they knew that whoever figured out the structure of DNA was going to get a Nobel Prize. So there were multiple labs trying to figure it out, and one group won. Some of the ones that didn't figure it out who were trying were Linus Pauling here in the US, but his model turned out not to be correct. But in England, we had the lab of Maurice Wilkins, where Rosalind Franklin worked, and they were using X-ray crystallography to study molecular structure. So they would dehydrate organic molecules to get them to form a crystal, and then shoot X-rays through it and figure out the structure of that molecule. So Franklin famously produced this image of a DNA crystal that had X-rays shot through it. And from this, I am not an expert at interpreting these images, but what she was able to deduce from her X-rays was that DNA was a double helix, and a helix is something that twists. So a double helix is like a ladder that twists. So double meaning there was two strands, and a helix meaning that it twists. And the phosphates and sugars were on the outside and the bases were on the interior. She did not figure out the exact structure. Now, she also did not get the Nobel Prize for her work. When the Nobel Prize was handed out some years later, she had already died of ovarian cancer, probably due to exposure to X-rays during her research. Now, at the time that the Nobel Prize for the discovery of the structure of DNA was handed out, the rule was that somebody who was dead could not get the Nobel Prize, because the idea was you're giving people money for research, and you can do research if you're dead. So Rosalind Franklin did not get the prize. Instead, Maurice Wilkins got it, and also Watson and Crick, who you probably heard of. So that rule was changed in 1972, and at any time after that, after 1972, Franklin could have been given the Nobel Prize posthumously in recognition for her work in the discovery of DNA. However, despite a letter campaign to the Nobel Committee that goes on every year, they could have awarded her the prize at any time since 1972, and they have not. So feel free to write them a letter on behalf of her. Watson and Crick were the other two Nobel Prize recipients. James Watson is an American. He was a postdoc working in Francis Crick's lab, and they figured out the exact structure. This is their model that they welded together in the lab. However, they would not have been successful without Rosalind's data. And they were given her data without her knowledge or permission by Wilkins. In science, it's considered unethical to share somebody's unpublished data without their knowledge or consent with competing scientists, which is exactly what Wilkins did. So many people have been critical of the Nobel Committee for not recognizing Rosalind Franklin's contribution at the time. And it's probably the case that if she had not died of cancer, that she would have gotten the prize along with the other three. But we don't know for sure if that's the case. Last time I checked, James Watson was still alive and living in California. Crick died decades ago. He was a smoker, and I believe he died of lung cancer or some other smoking-related cancer. So Watson and Crick took that information that DNA was a double helix and the bases were facing the interior and the sugars and the phosphates were on the outside, and they figured out the exact structure and how to make sense of Chargaff's rule that the amounts of A and T were always the same and the amounts of G and C are always the same. And they made sense out of that information. And their structure, Watson and Crick's structure of DNA, also suggested a mechanism for replication. Because if you always have A and T bound together, and you always have G and C bound together, then that means that the strands of the DNA are complementary. And we'll talk more about that. Okay, so here's our 4 nucleotides, G, C, A, and T, as we will call them. And so the entire nucleotide includes what's going to become part of that sugar phosphate backbone. So the part of the DNA molecule that's covalently bonded together. And remember, covalent bonds don't break when you dissolve them in water, like ionic bonds do. So here's our covalently bonded backbone that goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and that's stuck together. The bases kind of hang off of that, and they are going to stick together into the double helix by hydrogen bonding. So that double helix is like a ladder that can unzip for replication. And you'll also notice that there's a difference in the structure of the ends here, which we call the 5' end and the 3' end of our DNA strand. And this relates to the carbons in the sugar. So in organic chemistry, the carbons are numbered in many kinds of organic molecules, but in carbohydrates they are. So we have 1, 2, 3, 4, 5 here, numbering the 5 carbons in deoxyribose. And so the 5th carbon here, where that is going to take the next bond, that's the 5' end, and at the other end here where the 3rd carbon is going to take the next bond, that's the 3' end. And this is going to make a difference because during DNA replication, new nucleotides can only be added to the 3' end of a growing strand by DNA polymerase. So the X-ray images of Rosalind Franklin also showed that DNA had a constant diameter. So any model that Watson and Crick came up with would also have to take that into account, and their model did, in fact, account for that. So the strands of DNA, the 2 strands of the double helix, they are not identical. They are complementary. So every place we have a C on one strand, we have a G on the other strand. And where we have an A on one strand, we have a T on the other strand. That is complementary. And Watson and Crick's model also very neatly explained Chargaff's rule, that the amount of A and T are the same because they bind to each other. So they're always going to be in the same amount, and the same with G and C. If G binds to C, then the amount of those 2 is always going to be the same. And it's always nice when your model explains all the known data. That's important. So, and the reason why A and T bind together and C and G can bind together, they're hydrogen bonding together, and it's because of the way the electronegative elements and the positively charged hydrogens line up. Because remember, a hydrogen bond is between a partially negatively charged electronegative element and a hydrogen that is going to have a partial positive charge because it's bonded to something electronegative. So positive hydrogen, negative nitrogen, positive hydrogen, negative oxygen. And so we get hydrogen bonding. And these cannot bind to each other because the elements don't line up correctly for hydrogen bonding. And for hydrogen bonds, distance is really important. The distance between the atoms is important. So if they're just a little bit too far away, hydrogen bond will not work. So Watson and Crick realized that their model suggested a specific mode, a copying mechanism for the genetic material. They realized that because they're complementary, you could simply unzip the strands and then make a complementary copy of each one. Now, it took several more years to prove that this was actually what was happening. And we call this semi-conservative replication. So here's our original DNA molecule over here. And it unzips because remember, these are just hydrogen bonded together. So that makes separation fairly easy. We can make this happen in the test tube by just heating it up. That will break the hydrogen bonds. In fact, we do that during PCR and other kinds of DNA manipulation. So a complementary strand can be synthesized. So when DNA replicates, we actually end up with 2 molecules that are each one old strand and one newly synthesized strand, but they are identical. They are identical to each other, but this is semi-conservative replication. So DNA replication doesn't just start at one end and go along. It starts at specific origins of replication and moves in both directions from the origin of replication. And this proceeds a little bit differently in prokaryotes and eukaryotes. Prokaryotes have way less genetic material, so they generally have only a single origin of replication. But eukaryotic chromosomes may have hundreds or thousands of origins of replication. So here we have an E. coli. We have our single circular chromosome and a single origin of replication. Replication bubble forms. The two strands separate and the new DNA strand is being synthesized. It continues in both directions until two new complete molecules are formed. And here's a nice scanning electron microscope image of that caught in process where you can see the chromosome is partially replicated. For eukaryotic chromosomes, it's a bit more complicated because we have replication starting at many origins of replication and moving in both directions. And we are going to skip the portions in the chapter that talk about the details of this replication and the fact that because we can only have replication by adding to the 3' end of a growing strand, those are on opposite ends of a single double helix, which creates a leading and a lagging strand. So you can just skip all the stuff about leading and lagging strands and Okazaki fragments, and we're just going to skip that. There's a little video embedded in here in the PowerPoint, which you can watch. I'm not going to show it during this video. It's a couple of minutes long, but it's a nice little summary, so feel free to watch that on your own. So enzymes called DNA polymerases catalyze the elongation of a new DNA strand. Now, this is a case where the name of the enzyme tells us exactly what it does. So DNA polymerase, it polymerizes DNA from nucleotides. RNA polymerase makes RNA from nucleotides. And it's going to require, there's other things that are required for DNA replication. For example, because DNA polymerase can't start from nothing, it has to have a strand to add to. We are actually going to get RNA primers, and we're going to skip that stuff too. DNA polymerases vary between eukaryotes and prokaryotes as well. So prokaryotic DNA polymerases go much more rapidly, but they don't proofread, so they make more mistakes. So for bacteria, they can add about 500 nucleotides per second, and for eukaryotes, it's only about 50 nucleotides per second. So our DNA polymerase goes much slower, only about 1/10 the pace, but makes fewer mistakes. Which is good. So this is an uphill reaction. If you remember from our entropy and energy discussions earlier, a reaction that is creating one large molecule from smaller molecules is going to be an uphill reaction. You are decreasing entropy, which means we need energy. We talked previously about ATP being an energy source for many reactions, and GTP as well. Those are RNA nucleotides. But it turns out that the DNA nucleotides themselves, as we call them, the deoxynucleotides, they provide the energy for the DNA polymerase to make this reaction a downhill reaction. So the trinucleotides, remember the TP and ATP, this is adenosine triphosphate, we have those same ones, but the DNA version, and that's what the lowercase d there stands for. And we would always write them this way, lowercase d for deoxy, and then ATP. So deoxy ATP, deoxy CTP, deoxy GTP, and deoxy TTP. These are the DNA building blocks. We call them the DNTPs for short. all 4 of them together. So if you have DNTPs, that means you have all 4 DNA nucleotides in there, in your solution, whatever it is. So these are both the building blocks and the energy for DNA synthesis. As 2 of those phosphates are going to be removed, that's going to provide the energy for this anabolic reaction. And the same is going to happen for RNA, which we'll talk about RNA synthesis in the next section. So the 4 RNA nucleotides, which would be ATP, CTP, GTP, and UTP, would be the RNA nucleotides. They do exactly the same thing. Each nucleotide, as it's being added, 2 phosphates are removed, and that provides the energy for this synthesis reaction. So here's our double strand here, and we have our single strand extending down here, and our new nucleotide is going to be added. In this case, it's a T because A is over here. This is the reaction that DNA polymerase catalyzes. Remember, enzymes make spontaneous reactions happen more quickly. And it's going to be a spontaneous reaction because of the addition of energy. So just like ATP, any of the 8 nucleotides have, if they are triphosphate nucleotides, they have the energy here to help make a reaction, a spontaneous reaction. So 2 phosphates are removed, and that provides a little bit more energy than just removing 1 phosphate, and that provides the energy to create this bond here. So that's, and you notice it's adding to the 3' end. That's all DNA polymerase can do. It can only add nucleotides to the 3' end of an existing strand, which is why we need RNA primers to get it going, but we're going to skip that part. So DNA polymerase is our enzyme here. Each of the 4 deoxynucleotides provides the energy for this reaction. So why is there any error at all in DNA polymerase? Hasn't evolution been going on long enough that a perfect DNA polymerase couldn't have evolved by now? And the hypothesis is that has not been selected for, that having an error rate of 0 is not an advantage. Because remember that mutation is where new traits come from. If there was a mutation rate of 0, that would not be an advantage to that organism. So for eukaryotes, it appears that having a low error rate has been selected for because our DNA polymerase makes fewer errors than bacterial DNA polymerase. Bacteria have evolved for speed rather than accuracy. Our DNA polymerase goes much slower, and so our cell replication is much slower, but it doesn't make as many mistakes, but it still makes them. And these mutations then are one of the sources of mutation that leads to new traits that allow natural selection to have something to select. The ends of the DNA for linear chromosomes is a problem. So for bacteria and archaea that have circular chromosomes, they can just replicate all the way around. No problem. But eukaryotes have linear chromosomes. And here is another limit of the DNA polymerase molecule. So not only can it only add to the 3' end of a growing strand, it has to be sitting on the complementary strand in order to do that. And that's the problem, because when it gets out towards the end, it's going to drop off of the end of the DNA molecule before it completely replicates that other strand. And the amount of space that the DNA polymerase takes up is about 100 nucleotides, or 100 base pairs, we would call it. So that's about how much gets shortened every round. So every time the cell replicates, the telomeres, or the ends of the DNA, are going to get shorter. And so the telomeres, like the centromeres. The centromeres, if you remember me talking about the centromere, there's no genes in the centromeres. It's an area of the DNA molecule that's very repetitive. It attracts kinetochore proteins to bind there during mitosis and meiosis. The telomeres as well. There's no genes in the telomeres. They are the ends of the chromosomes. They have just repetitive sequences in them. And they get shorter every time the cell replicates. And this is the reason for the Hayflick limit. The Hayflick limit for human cells is about 50 divisions. That's about how many divisions it takes to erode the telomeres. Once the telomeres are gone, a normal cell will stop dividing. Now, there are cancer cells who will keep dividing even when the telomeres are gone, but that's another story. And it's also considered to be connected to aging, because when you take tissue samples from people of different ages, older people have shorter telomeres than younger people. And there are various environmental things that have been shown to shorten telomeres, like stress. So if this happened in every single cell, though, that would be bad. So germline cells, of course, we want them to have full-length chromosomes, or the species would go extinct very quickly. So germ cells have an enzyme, another DNA polymerase called telomerase, that it can't replicate just any old DNA. What it does is make telomeres. So it will re-lengthen the telomeres every time the cell divides. And so germline cells have telomerase, and every time they divide by mitosis, every time the primordial germ cells divide by mitosis, or spermatocytes divide by mitosis, I guess that would be spermatogonia, divide by mitosis, the telomerase is going to re-replicate the telomeres. There are some stem cells as well that have telomerase and don't lose their telomeres and can divide more than 50 times. So telomerase is a DNA polymerase. It's just a very specialized one that can only make those repetitive DNA sequences at the end of the chromosomes. Okay, so the beginning of this section, we talked about experiments with phages that provided the final proof that DNA was the genetic material, using radioactive sulfur and radioactive phosphorus. DNA was finally figured out to be a double helix with two anti-parallel strands. So the 5' and the 3' ends go in opposite directions on those two strands. Replication is semi-conservative. We are not going to worry about all the details about DNA replication. There are many enzymes involved. We're just going to worry about DNA polymerase. The energy for this reaction comes from the nucleotides themselves. All 4 of the DNTPs provide the energy. DNA polymerase there are several DNA polymerases, are the enzymes. Telomeres shorten every cycle. That's the reason for the Hayflick limit. Telomerase is another DNA polymerase that's present in only certain cells, like germline cells and some stem cells, and that re-lengthens the telomeres so that those cells can divide more times than the normal 50 that would be expected. All right, and we will go on to talk about RNA and gene expression. All right, so let's talk about RNA now. RNA, I think, is more interesting than DNA. DNA does one thing. It's the genetic code. But RNA is really doing all the work of gene expression. We'll talk about messenger RNA, which is involved with taking the code of the gene from the nucleus out into the cytoplasm to the ribosomes. tRNA and rRNA, which is involved directly with translating the code from the code of nucleotides to the code of amino acids and proteins. And we'll talk a bit about small nuclear RNAs as well, which are involved with preparing the messenger RNA. So we used to have the one gene, one protein hypothesis in the 60s and 70s when the genetic code was first being worked out. And what this meant was that it was thought that every gene coded for one single protein. And now we know that some genes code for more than one protein, slightly different proteins, and one gene can also code for an RNA, and that often RNA is the last product of that gene. It doesn't go on to form a protein. Like the tRNAs and rRNAs themselves have their own genes that code for them. So now we often say gene product because that would include not just polypeptides or proteins, but also RNAs if those are the final gene product. So we need to define two words here, transcription and translation, very important in gene expression. Transcription means one thing. When you hear something in the cell, there's some kind of transcription going on. It means we take a DNA and make an RNA copy of it. And that's really step one in gene expression. So when a gene is expressed, that means it's being turned on. And whatever that gene codes for is going to be made. That's gene expression. So the first step in that is transcription. And we use the word transcription very deliberately here. So a transcript is an exact copy. If you have a job transcribing doctor's notes into the computer, you're making an exact copy. So that's what we're doing with RNA. RNA is made of nucleotides, DNA is made of nucleotides, and RNA copy is a nucleotide for nucleotide copy of the DNA. That transcript, if it's going to make protein, is called a messenger RNA because it's taking the message of what that gene codes for out into the cytoplasm. Or if it's a bacteria that doesn't have a nuclear membrane, then it's just taking it to the ribosome. Translation is the next step in the process. Translation is taking that RNA sequence and turning it into an amino acid sequence. Because if you remember from our biochemistry, a protein is a sequence of amino acids. It's a linear sequence of amino acids. So translation is taking that RNA sequence and turning it into an amino acid sequence. And we use the word translation here because we're changing languages. If you translate something, it means you're going from one language to another. Translation here means we're going from the language of nucleotides to the language of amino acids. So that's why we use the word translation here for this process. And translation occurs at the ribosomes, which we'll talk about those later in this chapter, exactly how the ribosomes accomplish this. So here was our central dogma of one gene, one RNA, one protein, and this was called the central dogma of genetics. And we would now modify this to say one gene could produce several different messenger RNAs, or one gene can produce an RNA as the final product of that gene. If, for example, if it's a tRNA or a ribosomal RNA, that's the final product. A messenger RNA can be translated into a protein, but that's not necessarily the final gene product. An RNA could be. So in a bacterial cell, and when we say bacteria in molecular biology, we usually mean an E. coli if it's not specified which type of bacteria that is. So transcription, the first step, we're going to make a RNA copy of a gene. That's the messenger RNA. Translation, the next step, that messenger RNA will attach to a ribosome and it will be translated into a polypeptide. And that's the next step. So we're going to talk about these steps in more detail. So in prokaryotes, prokaryotes, remember, means things that don't have a nuclear membrane, like bacteria and archaea. In prokaryotes, translation and transcription can occur almost simultaneously. So translation can start after transcription has begun, even just a little bit, because there's no nuclear membrane separating the ribosomes, which in a eukaryotic cell are out in the cytoplasm. There's no nuclear membrane separating the ribosomes from the newly formed transcript that's being made, the RNA transcript that's being made from a gene. So translation can begin. This is a nice image here of you can see the DNA, this very thin structure here, and these teardrop shaped blobs here, those are the RNA polymerase molecules. And just like DNA polymerase catalyzes the formation of DNA from the nucleotides, RNA polymerase does the same thing for RNA. So RNA polymerase takes individual RNA nucleotides and links them together into a long RNA strand. And just like for DNA, the energy for this reaction comes from the nucleotides themselves. Each trinucleotide, 2 of the phosphates are removed, and it is added to the sequence. That provides the energy for this reaction. Okay, so here we have our DNA chromosome here across the top, and the small molecule, see the little tiny black structure? That is the RNA polymerase molecule. So it's a large polypeptide. And then these larger black blobs on here, those are ribosomes. So here we have from a bacterial cell. And of course, this isn't in a cell right at this moment. This was smashed out on a slide so that we can see an image of this. But you can see that translation is occurring at the same time as transcription. So these little RNA polymerases are moving along down the DNA. As they do, they are making an RNA copy, which you can see diagrammed down here, RNA copies of that gene. And we can tell that RNA polymerase is moving in this direction because of how long the RNA is down here. So this is much further along in transcription down here. And the reason we can't see any polypeptides emerging from each of these ribosomes, so each ribosome has a polypeptide emerging from it. And the reason we can't see that polypeptide is because it's smaller than the resolution of this image. Even though DNA is quite skinny, because it's associated with protein, we can see the DNA here. It looks thick enough, we can just barely see the DNA. But the polypeptides that are coming out of each ribosome are too small to be seen with the resolution of this particular image. So that's in prokaryotes. Now in eukaryotes, that can't happen because we have a nuclear membrane. The DNA does not go out into the cytoplasm, and functional ribosomes are not inside the nucleus. So transcription occurs, and you'll notice we have this different description here. It says pre-mRNA, and that gives you the idea here that something else is going to happen to this messenger RNA before it's ready to go to the ribosome, and that's exactly what's the case. There is some RNA processing that's going to occur. And we'll talk about that in detail. In fact, there's 3 things that are going to happen to this messenger RNA to allow it to function and be translated once it gets out into the cytoplasm. So the pre-messenger RNA, sometimes this is called the primary transcript or the initial transcript. So the RNA that's made from transcription is called the transcript. And the initial transcript, that would be the first transcript that gets made. So that's our pre-mRNA here. Now, that primary transcript, the initial RNA transcript, is the copy of the gene. And there are other regions in that RNA other than the exact sequence that's going to be translated by the ribosome into a protein. There are other regions in that transcript that are important. UTR here stands for untranslated region. So these are not translated, meaning they don't contain the genetic code. So we have the head end of our RNA is the 5' end, and the tail end is the 3' end. If you remember from DNA, DNA also has this 5' and 3' end that has to do with the which way the ribose is facing, and RNA does too. And so the beginning of the transcript is always going to be the 5' end, and the tail end is always going to be the 3' end. And like DNA polymerase, RNA polymerase adds to the 3' end of a growing strand. So that's why the 3' end is the tail end. Those are the last ones being added. And you'll notice there's 2 untranslated regions here, a 5' untranslated region and a 3' untranslated region. And that just describes which end it's on. So the coding sequence here, and we'll talk more about the coding sequence in a few minutes here. The coding sequence, that's where we find the genetic code. This information, the coding sequence, is where we find the information to make that amino acid sequence. Which amino acids are going to go into that polypeptide? The 5' untranslated region and the 3' untranslated region also contain important information. One of the things that's contained in the 3' untranslated region is sequences that help control this messenger RNA and determine whether it's going to be digested and broken down and not used anymore. And then you'll notice we also have a cap and the poly A tail, which we'll talk about that. Those are additions to this process. Okay, so for an overview here, so we have our DNA in a eukaryote. Transcription means we're going to make an RNA copy of that, and the initial copy is just a nucleotide for nucleotide copy. It is going to be processed, and there are 3 things that go into that processing, which we will talk about in detail. Then it is ready to go out into the cytoplasm. It leaves the nucleus through nuclear pore, and then translation occurs at the ribosome. So you can see that one big difference here is that both things cannot occur simultaneously like they do in a prokaryotic cell, which means it's slower. So in a prokaryotic cell like a bacteria, they can respond rapidly to changing environmental conditions, because as soon as a gene begins expression, as soon as that transcript starts to be made, they're already starting to make the proteins. They can respond to environmental changes very rapidly. Eukaryotic cells are slower because of this barrier of the nuclear membrane and the RNA processing that has to occur. So slower to respond to environmental changes, but more fine-tuned in the ability to regulate the amount of protein that's going to be made from one of these genes.